So thank you first for inviting me to this exciting event. My name is Jan Lotvall and I'm a professor of allergy at University of Gothenburg in Sweden, where for many years have been working in the field of asthma and understanding asthma disease, as well as testing different types of drugs in asthma primarily. About 20 years ago, we were starting to look into whether exosomes, also called extracellular vesicles, could be involved in the mechanisms of asthma. And that research was not so easy, and we were very close to quitting that research when we discovered that they have RNA-containing proteins. They carry RNA-associated proteins. And that made us think maybe they have RNA as well. And we discovered RNA in the exosomes and could show that the exosomes send RNA from one cell to another cell, which at that time was crazy. It was thought that RNA only stays inside the cell where it is produced. So this was kind of a new concept, and it changed my focus of research. I continued working on asthma. I still do some asthma research, but the focus had to become these exosomes. And of course, the work was highly cited and recognized, and that has given a lot of attention to the field as well, which is just an honor. But since then, I want to emphasize, since then we have learned so much through so many years of hard work by many students and postdocs. So it hasn't been an easy journey. Fun, but it's not easy. Clearly, exosomes, extracellular vesicles, are not so strange, actually. They're very prominent in our bodies. Our cells produce large quantities of them all the time, and there's communication going on between all cells at all stages of life. Primarily, we see that immune cells such as macrophages and dendritic cells are prone to take up the extracellular vesicles. And that means that if you want to develop a drug or a treatment, you should try to target diseases that are associated with those cells, macrophages or dendritic cells, which are immune cells, and inflammation can be targeted in that way. So that's why the anti-inflammatory regenerative exosomes are interesting, and they are primarily produced by the mesenchymal stem cells in the body. You have 2 billion mesenchymal stem cells in your body, and they produce maybe 1,000 or 10,000 exosomes every day. So you have trillions of exosomes, but you have them spread throughout your whole body. So that's an issue. If we really want to get the drug to the right place at the right time, you need to have a high concentration and a different way of administering them. So the exosome, obviously all cells produce exosomes, and not all cells are regenerative. A minority of them are, not all of them, because we have so many more than we have cells. But these exosomes that come from mesenchymal stem cell have this anti-inflammatory and regenerative effect. And it's our ability to isolate such cells, expand them in cell culture, and then harvest those exosomes. It makes it possible for us to give the exosomes in a high concentration to the patient at the right time, at the right dose, and at the right location. So that's what's special about mesenchymal stem cell or stem cell exosomes in general. So clearly, there's already use of exosomes in cosmetics and cosmeceuticals, and it's being quite successful. But the end goal for me and many other scientists is that we can use them also for treatment of disease. And of course, skin disease, inflammatory skin diseases is one possible target, but also other diseases such as severe lung injury or inflammation in the lung could be targeted with exosomes from stem cells. And that's actually been done. There were several studies during the pandemic, during the COVID pandemic, where patients were giving these exosomes either by blood or by inhalation, and you could see very clearly anti-inflammatory effects and even life-saving effects of these exosomes. So I think there are two things that have happened over the last 20 years that makes it a little bit easier, still difficult, but a little bit easier to develop them as drugs today. And one is we understand them better. we understand which exosomes have this anti-inflammatory function, for example. And the other thing that is really important is manufacturing. And of course, when you manufacture for treating a disease, you need to have what's called good manufacturing practice. You need to have a very large scale, because these are expensive little magical bullets for treating disease. So that is another aspect that is very important and makes it easier for us today to be able to translate the science to real clinical practice and real clinical treatments. And if you want to develop a drug, it has timelines. It always have timelines that are at least 3 years or maybe 4 years. If you're developing other types of products, such as food products or skin products that are not medicines, it's of course much easier and has a shorter timeline. But what I think is crucial is that you understand your manufacturing process, you understand the quality attributes of the product that you're making, and you know the safety of that it doesn't have viruses or bacteria or anything like that. Those are some of the crucial aspects to consider when you want to develop products quickly. So there are two angles of this question. And one is, why is exosomes especially suited for the skin? I think they actually are. And that is because you can apply them to an area exactly where the disease is without any complicated sort of way of administration. Now, if you give to treat COVID-associated lung injury, for example, you need to give it by inhaled route or by infusion, and not all of the drug will end up exactly where it should go. So from that perspective, skin disease is more suitable. There are many other types of drugs that can be developed for skin that also are prone or effective to give by local administration, but this is where kind of exosomes have a unique opportunity and an early opportunity, in my mind, for real drug development. Yeah, so one of the fundamental aspects of these mesenchymal stem cell exosomes is that they inhibit inflammation. And especially they inhibit and attenuate, slow down the inflammation that is mediated by bacteria or viruses, or what we call innate immunity. It's also associated with cell death and damage and stuff like that. Corticosteroids, for example, are not as effective as mesenchymal stem cell exosomes are for treating those diseases. So I think we have an opportunity to really be able to treat aspects of diseases that we don't yet have medicines for. So that's a challenge to convince regulators on how to achieve that. But of course we have to, because patients have to have access to these new medicines. Fundamental requirements. One is potency and efficacy, right? They have to be effective in the disease you're treating, and they have to therefore reach that disease. And if you're giving it topically or by local injection, then into the skin, then you would do that. And the other key attributes is always safety. So good source of the exosomes, which in this case is a stem cell without any viruses or anything like that potentially could come with the exosomes. And of course, they have to have a good, stable product that we know can be distributed in an efficient way and reach the customer and still being functional. So there are a number of complicated aspects to consider. It's just not what we do in a research lab. We take some exosomes, we put them with a pipette into a cell culture medium, and then we see something. Making a drug is much more complicated. When you say exosomes, you should say stem cell exosomes, because there are 99.999% of the exosomes we have in our bodies are not stem cell exosomes. They're just normal cell released by any cell, like the red blood cells or endothelium or anything, right? So exosomes is not one. The origin exclusion, the way they are administered is crucial, and the key attributes is that they have to have this biological function that you expect to have, right? They have to be repairing to the skin. You have to show that in different models. A lot of these things can be done in cell cultures these days, and that's very good. And they have to have this anti-inflammatory function that they so uniquely have. So I think exosomes have a big future far beyond regenerative skin treatments, right? It's going to be a drug. It's going to be a drug against severe inflammation. It's going to be a drug that can target certain cells in the body. We will probably have exosome drugs that can deliver certain medicines in a more effective way, maybe deliver insulin in a more efficient way instead of injecting it subcutaneously, having a pump. So there are so many opportunities here. I often compare with monoclonal antibodies. They were discovered in 1975 in Oxford. And now we're seeing every year new types of monoclonal antibodies that function as biological medicines. They're very, very expensive to treat, but we can cure cancer with some of these monoclonal antibodies called checkpoint inhibitors, right? They can help the immune system to kill the cancer. And there are a whole series of those type of medicines that are still coming out from a discovery that was done in 1975. And what's happening today in 2025 is that we are starting to find ways of using these things as medicine. And in 2075, we will have a massive amount of exosome therapeutics for all kinds of diseases that are more effective than the biologicals we have today. So then you have to use a totally different type of exosomes. We have some research into trying to treat cancer, but in that case, we don't use stem cell exosomes. We use bacterial exosomes, and we engineer the bacterial exosomes to help the immune system to target specifically the cancer. So if you ask me that, I would say yes, I think so. It's hard to get investments and pursue these developments, but I think there's definitely an opportunity to create therapeutics that will help cure cancer in the future, yes. So when you look at the scientific literature about exosomes, a large majority is based on cancer studies of how cancer cells behave and experimental models of cancer. And therefore, the title Exosomes and Cancer are often together in the title of the paper. And therefore, because people think exosome is one thing, they think it's also associated with cancer, but this is fundamentally wrong, right? So if you're working with cancer exosomes, they could do things in cancer. They can help promote their own cancer. They can help the cancer to penetrate tissues and migrate into tissues, metastasize, and so on and so forth. Now, totally different exosomes, such as stem cell exosomes or even bacterial exosomes, do not have those sort of features. So I cannot understand or see how a mesenchymal stem cell exosome could induce cancer. Another thing is that people have tried to create cancer by using cancer exosomes. Take those exosomes and put them in another cell and and that's extremely hard and basically has not been convincingly proven that it is even possible. Not even a cancer exosome can seem to be able to do that. So I don't think it's an issue. So when you talk about plant, you should distinguish plant exosomes, which is often extracts of plants, and plant stem cell exosomes, which is something fundamentally different. It's a stem cell that releases the exosomes in a cell culture, and then you isolate them and purify them and use them in your product. And that's possible to do in the skin. So I think there is a lot of work to try to understand and distinguish the different plant cell exosomes. But I'll be totally frank, I think the big future is with normal human stem cell exosomes, and exosomes that are produced with advanced engineering of other cells, creating basically stem cell-like exosomes by engineering. And it's happening right now. And thank you very much for this interesting discussion. I enjoyed it. Thank you.